Renelle's is joining us via my phone, so I'll share her presentation. So in a sec, you'll see the presentation and hopefully you'll hear Renelle.
Okay, thank you, Steve. I hope we can make this work. So let's start by how do we unlock our rhizosphere's full potential? I want to open with a statement. A plant's rhizosphere is a microbial hotspot and is considered one of the most complex ecosystems on Earth. I think all of us are in agriculture, and what a privilege to work with one of the most complex ecosystems on Earth. I always thought the most complex ecosystems are in the Amazon or someplace exotic. And yeah, we have the opportunity to work with one of the most complex ecosystems on Earth. So just by opening with this statement, I want to say everything from here on is oversimplified. So we've learned a lot about the rhizosphere and the microbial interactions, but there's still so much more. So the rhizosphere, or the root surface, is that 5 millimeter surrounding the root. It's also the area that's under the direct influence of the root exudates. Next page.
Right.
Yeah. Okay. So it's also the area that's under the direct influence of the root exudates. In other words, the carbon that's secreted by the root to feed the microbes in the rhizosphere. The microbes in the rhizosphere produces a biofilm. First of all, to protect these rhizosphere microbes. You can also see on that picture, the rhizosphere has a very unique pH. So that is a picture of a corn root system. So you can see the pH of the rhizosphere is more or less 7, whereas the soil pH is 5.5. So the environment in the rhizosphere, the whole chemical composition and environment there is totally independent and different from the bulk soil. So it's a totally independent environment. And that's also why the rhizosphere is crop specific. So if I put up a legume rhizosphere here, you would see it's a totally different pH. There's obviously different root exudates, so it's chemically different. So every crop has a unique rhizosphere, and because of that unique group of microbes that establish in the rhizosphere and interact with the plant. Next. Okay, so life in the rhizosphere. The rhizosphere is literally established within the first few days of a plant's life. And then it is a lifelong relationship or synergism between the microbes in the rhizosphere and the plant. So this is also a dynamic community of microbes, a dynamic relationship. So the rhizosphere community changes over the plant's lifetime. So you might find certain groups very prominent in the beginning of the plant's life, stimulating growth, stimulating certain functions in the plant, photosynthesis, and later on they will almost disappear from the rhizosphere and other groups will become more prominent. So the rhizosphere is also majority bacteria, just like the bulk soil. And if we look at the signaling in the rhizosphere, there's a three-way signaling or communication between the microbes and the plants. So first of all, there's a plant to microbe signaling. So how the microbes communicate with the plant, then how the plant communicates back to the microbe, and then how microbes signal to each other. Okay, so first we're going to look at how the plant communicates with microbes. So plants use metabolites from primary metabolism, for example, organic acids, carbohydrates, amino acids, basically carbon sources and amino acids, nitrogen sources that it secretes to tell microbes there is a source of food available. And that is the primary signal to recruit microbes to the rhizosphere. Also some secondary metabolites, for example, alkaloids, terpenoids, phenolics, for example, the flavonoid involved in the signaling between the plant and the rhizobia, telling the rhizobia that it's time to infect and form nodules. So plants use these metabolites or signaling molecules. First of all, to tell microbes here is a source of carbon, so they come to the rhizosphere and they establish this synergistic interaction. Then also it signals its needs. So what's going on with the plant? It's in an optimal state of health, or there's stress in the environment, or there's a pathogen invasion. So everything that's happening with that plant is communicated via metabolites to the rhizosphere microbes. And then also to interfere with the rhizosphere microflora. As I mentioned, the rhizosphere is dynamic, so it changes the whole time depending on the plant's needs. So all of these microbes in the rhizosphere obviously work for the better good of the plant. So they are there to keep this plant in optimal state of health and photosynthesis, because then is when the plant secretes their share of the carbon coming from photosynthesis. So plants use also these signaling molecules to establish plant-specific microbiomes. So every plant has a unique microbiome or a unique environment and community of microbes in its rhizosphere. Okay, so let's look at microbe to plant signaling. So plants use hormones, or microbes use hormones, for example, jasmonic acid, ethylene, salicylic acid, growth hormones, to induce specific genes inside the plant. So these genes are usually involved in the plant's immune system or its resistance towards infection. Then you also have molecules on the surface of microbes. I call it the microbe's fingerprint. And these molecules are signaling molecules that tells the plant this is a beneficial microbe, allow it into the rhizosphere or into the endobiome, or this is a pathogen, launch an attack. So it's very important for a plant to be able to distinguish based on these MAMs or microbe-associated molecular patterns if a microbe is part of the beneficial group of the rhizosphere or something must be done to keep it out. Okay, and then lastly, microbe to microbe communication or signaling. So microbes also communicate with each other. So there's no single group or single microbe in the rhizosphere that is on its own mission, doing its own thing. So all of the microbes in the rhizosphere are in communication with each other, responding to signals from the plant based on its needs, and then collaborating to fulfill these needs. So the signal molecules that microbes use switch on genes in other microbes. So they actually activate genes and metabolic pathways in each other. They also use these signaling molecules to attract microbes for a specific function. For example, we call it chemotaxis. So chemotaxis is just a chemical signal that is sent out to get microbes to move into a specific area and perform a specific function. Yep. So bacteria use these signaling molecules to coordinate their movement. They move together, they grow together, they coordinate their metabolism, and they also coordinate their biochemical processes. So one will focus, for example, on nitrogen fixation, a different group will focus on phosphate solubilization. So they coordinate all the biochemical processes based on what the plant's needs at that stage is. Then the second group of microbes that are also very involved in signaling are endophytes. So endophytes produce signaling molecules, first of all, to get microbes with a specific function, as I mentioned, nitrogen fixation, phosphate solubilization, maybe pathogen suppression, to recruit those groups, those functional groups that we call them, to the rhizosphere. And then also to establish these beneficial interactions between the plant and the microbe. So let's compare the microbes in the rhizosphere, that is plant associated, with the ones living in the bulk soil, the free living microbes that are not plant associated. So the rhizosphere microbe lives in the rhizosphere, it's in a direct interaction or symbiosis with the plant, whereas the free-living microbe is living in the soil, no direct interaction with any plant. The rhizosphere microbe gets its carbon and nutrients from the plant, whereas the free-living one the soil is its source of carbon and nutrients. The rhizosphere microbe is obviously important in plant nutrition and health via the production of enzymes, metabolites, and all these other signaling molecules, whereas the microbe that is not plant associated is part of the soil food web in the bulk soil. So totally different dynamic that's going on there versus the rhizosphere. And then also rhizosphere microbes are very important in plant health via that induced systemic resistance, the molecules they produce to switch on certain genes that give the plant better resistance towards pathogen infection and stuff like that. Whereas the free-living microbe is very important in soil health, for example, suppression of dominant pathogen groups in the bulk soil. So the main difference between a rhizosphere microbe and a free-living microbe in the bulk soil is its focus. A rhizosphere microbe is focused on the plant. Everything it does is to the benefit of the plant, to keep the plant in an optimal state of photosynthesis, to keep the plant healthy, to prevent pathogen infection, because only then they receive their part of the photosynthase. And up to a third of the sugars produced by photosynthesis can be secreted to the microbes in the rhizosphere. Whereas a free-living microbe, they live outside in the soil. They have no direct interaction with plants. They're part of the soil food web. The soil is their source of food and provision, so they're part of a totally different environment. So the role of rhizosphere microbes. First of all, the moment microbes establish in the rhizosphere, they start producing a biofilm. And the biofilm is first of all there to create a concentration of microbes, but also of metabolites. So it impacts the efficacy of those rhizosphere microbes. So that biofilm that concentrates the microbes in the rhizosphere and also concentrates the metabolites are there first of all to get better efficacy, but it's also a physical barrier. So the moment the biofilm has been established, it is the first line of defense against pathogens in the soil. Microbes produce a wide range of metabolites, as we discussed before, some of them signaling molecules, but also enzymes, for example, cellulases, glucanases, chitinases with specific functions that produce these signaling molecules that we've discussed, jasmonic acid, salicylic acid, also plant hormones that are involved in growth and signaling, and then also antimicrobial peptides. So peptides responsible for antifungal activity, some to suppress bacterial pathogens, others to keep out viruses, and also insecticidal toxins. So to summarize, a rhizosphere microbe is actively involved in every aspect of plant nutrition and plant health. So these microbes are really there to the benefit of the plant. It's there to keep the plant healthy and an optimal state of photosynthesis in order for them to get their third of the photosynthate secreted through the roots, to the rhizosphere. Okay, so we've discussed the free-living microbes in the bulk soil. They are not plant associated. The ones in the bulk soil that respond to the signals from the plant, those carbohydrates that it secretes and are the signaling molecules saying here is a source of carbon and of food, those we call rhizosphere competent. And then you get one step further, the endophytes, that not only establishes in the rhizosphere, but also enters the plant tissue and forms a very close interaction with these plants. So the free-living microbes, as we discussed, is part of the soil food web. The rhizosphere competent microbes that have responded to the signals from the plant and established in the rhizosphere, they are part of the functional groups in the rhizobiome or the rhizosphere team, I call them. So they are there for a specific function. And then the endophytes, if we look at what their function is, they are very involved in signaling communication with the plant, but also communication with the microbes in the rhizosphere. So we call them the coaches. They are the coaches of the rhizosphere team, planning the strategy to keep this plant in an optimal state of photosynthesis and health. So the advantages of a healthy rhizosphere, obviously it's a lifelong interaction. So as long as the plant is photosynthesizing, there's some carbohydrates secreted to the rhizosphere microbiome and they fulfill their function. So these microbes induce genes and metabolic pathways in the plant. So we know from scientific experiments that the genes that they switch on in the plant are involved in the uptake and utilization of nutrients, genes involved in the uptake and utilization of moisture, genes involved in how the plant will respond to stress, and also just the plant's general immunity or resistance towards disease. So they are all involved in switching on these genes and upregulating them if necessary. And then also these microbes in the rhizosphere are very involved in the energy economy or the productivity of this plant. And that in the end translates to yield. So the plant must have obviously enough energy for its metabolic functions, but also to secrete to the microbes in the rhizosphere. So the microbes will do anything to put this plant in a better energy economy to give a bit better yield, but also to support them. And a very interesting one for me is the microbes in the rhizosphere are actually involved in switching on genes, for example, for antioxidants in the end product. So a healthy rhizosphere not only gives you yield, but it also determines the quality of the end product, for example, the antioxidant levels. So these days we have very valuable tools to study rhizosphere microbes and endophytes, and also what's happening genetically in the plant. So we can look at the DNA of these microbes and the plant, and also the RNA. So DNA is just telling you the potential. So for example, this specific microbe in the rhizosphere has a gene that looks like it could suppress nematodes. And the moment that the gene is switched on, it produces an RNA molecule that tells us this microbe is now actively producing a toxin to suppress nematodes. So the one is just potential and the other one is telling you what is actually switched on at that moment. So endophytic trichoderma, literally trichoderma living inside the plant. So I refer to them as the trichoderma sitting in the control room, and they play a very key role in signaling. As I mentioned, signaling first of all, receiving the signals from the plant, communicating the plant's needs to the microbes in the rhizosphere, and also making sure that all these functional groups perform their specific function to contribute to this team. So it's really coordinating this team in the rhizosphere. So the coach sitting in the control room, coordinating the team in the rhizosphere, and also signaling back to the plant. One of the major roles of the endophytic trichoderma is to switch on genes involved in photosynthesis. So it actually upregulates the photosynthesis in the plant in order for the plant to be able to afford this whole team in the rhizosphere. Okay, so this is just a visual of what is happening with the endophytic trichoderma. The coach sitting there recruiting all these functional team members to the team. So we've seen with this specific strain of endophytic trichoderma, there's always a group of streptomyces. So we could see from the DNA and RNA, this group of streptomyces is involved in plant growth promotion, but also producing molecules keeping bacterial pathogens out of the rhizosphere. Then there's a group of Azospirillum, involved in free-living nitrogen fixation, not like the rhizobia infecting the plant, but just sitting there in the rhizosphere and binding nitrogen from the atmosphere and feeding it to the plant. Then there's a group of bacillus that we could see from the genes are potentially involved in keeping out nematodes from the rhizosphere. And then another group of streptomyces also involved in plant growth promotion, but also keeping out fungal pathogens. And then the final group that we've been able to identify are the Pseudomonas group involved in phosphate solubilization. In other words, phosphate that we know is it's the building blocks of ATP, and ATP is the energy currency in the cell. So the energy status of that plant depends on a functional group like Pseudomonas solubilizing, producing these organic acids, solubilizing phosphate, and making sure that it's converted to ATP or energy in the plant. So the endophytic trichoderma and its team obviously work towards one common goal, and that is to keep the plant in an optimal state of photosynthesis and health. Because the moment the plant stresses, the moment it's energy deficient, the first the supply to the rhizosphere is cut. So this team knows it. So they will do anything to keep the plant in an optimal state of photosynthesis and healthy, because the moment there's a stress signal or the plant experiences a pathogen attack, the carbon supply that goes to the rhizosphere is turned off. So let's look at the different functions as I discussed. All the microbes in the rhizosphere perform a specific function. So the functional groups that we've been able to identify in the rhizosphere is obviously first the endophyte that is involved in very central to signaling between the plant and the rhizosphere microbes. Then you get the bioinsecticidal group in the rhizosphere, keeping out insects. Then you have the free-living nitrogen fixation group involved in binding nitrogen from the atmosphere, but also involved in the nitrogen use efficiency of the plant. For example, how the plant will convert inorganic nitrogen to amino acids and proteins. And then there's the biofungicidal group, keeping out fungal pathogens. The group responsible for keeping out bacterial pathogens. And then also, as we mentioned before, the group involved in phosphate solubilization, but also the conversion of that phosphate into the energy molecule ATP. So what does the untreated rhizosphere look like? What's the most likely situation if you don't have a beneficial coach in the rhizosphere? So the opposition of the beneficial coach is usually an endophyte, a pathogenic endophyte like Fusarium. And Fusarium also puts together his best team in order to perform its function. So the members that we usually see grouping with Fusarium is Pythium, that we call a nibbler, making wounds, nematodes, Rhizoctonia, also nibblers, and then Phytophthora, killer. So Fusarium and its team, or some refer to them as a disease complex, also work towards a common goal, and that is to obtain sugars from cellulose. And cellulose is dead plant material. So they see a plant that's stressed as weak genetics, take it out and convert it to sugars and to carbon for the bulk soil, the soil food web. So they're actually doing the recycling. So if we look at soil-borne pathogens, are mostly fungi and nematodes. They live in the bulk soil of dead roots and crop residue. They can survive unfavorable conditions because they produce survival structures like spores or sclerotia in the case of fungi. And nematodes use a process called anhydrobiosis. So they literally dehydrate themselves and become dormant. And the moment the moisture situation is better, they become active again. And they also are stimulated by seed or root exudates, the same as the beneficial ones. So the root exudates is the signal for there's a food source. And then, as we discussed in the previous slides, you get two groups of pathogens, the nibblers making the wounds, and then the killers coming in, first of all, damaging the roots so that the plant can't take up nutrients and moisture anymore, and then also blocking the vascular tissue, taking out the plant. So this was a very interesting trial that we did just to prove a specific concept. So we were saying, obviously a farmer that is focused on soil health have a very good diversity of microbes in the soil. So if you put an endophytic coach on that germinating seed, it will recruit a very good team of rhizosphere groups to the rhizosphere, whereas a farmer that is not very focused on soil health and practices that benefit soil health will probably have a weaker team in the rhizosphere. You can put on the coach, but I mean the coach can only recruit what is available in the soil. So in this situation, we inoculated the coach, the endophytic trichoderma, and we're looking at what is the best available team it could put together from what is available in the soil. So there we saw a yield increase of up to 17%, which is very surprising. And then we went one step further and said, okay, we're not going to depend on the best available team in the soil. We're going to give the coach a team. So we're going to give him the functional groups that we know it needs to perform its function. So the phosphate solubilizer, the nitrogen fixer, the group that suppresses fungal pathogens, the group that suppresses insects, the group that suppresses the bacterial pathogens. And there you can see the yield response up to 28%. So this is obviously one trial at one site, 1 year. So it's not it's a statistical trial, but I mean we're just looking for a trend. So we're very excited to really repeat this trial at different locations with and see how we can get closer to the truth. We also did it on soybeans. On soybeans, it was also interesting. We not only treated it with the coach and waiting for it to establish the best available team, we also gave it a single dose of rhizobia. So this was virgin soil, and we gave it a single dose of rhizobia, and it put together its best team. So there you could see a 20% yield increase. Then, because it's virgin soil, we know it's a numbers game. So we gave the coach a double dose of rhizobia and also see what's the best team it could put together. So there you see the yield increase suddenly going up to 63%. And then in the final one, we give the coach not only a single dose rhizobia, but the A team, the team that consisted of all those functional groups, and see what the yield increase is at. So there's definitely truth in the statement that virgin soil is a numbers game. So we saw that yield increase when we gave it the double dose rhizobia, but also in the case where you have all the functional groups, the single dose rhizobia was sufficient. Okay, so there's basically two strategies when you use an endophytic trichoderma or a coach. So the best strategy is always to use the endophytic trichoderma at the establishment of the rhizosphere. Then you can use the product at the recommended dosage. You know that trichoderma is going to recruit the best team that's available in your soil, and the trichoderma at its best team is going to fight against the pathogens, keep them out of the rhizosphere, keep the plant in an optimal state of photosynthesis and health. So that's the best line of defense. The second best will always be to introduce the endophytic trichoderma after the rhizosphere has been established, especially when there is disease symptoms present. So then you have a situation of biological warfare. So now you have to use a double dosage of that endophytic trichoderma in order to, first of all, be able to enter the rhizosphere, fight off the pathogens, and establish a new team in the rhizosphere. So initially, it's only the trichoderma getting in, fighting off the pathogens, and then it takes time to establish a beneficial team in the rhizosphere. Okay, so this slide is just a photo of a DNA chip from a corn plant. So every dot on that photo is a gene that is switched on. So there's 3 sets of genes that we could see. The first ones are the green dots. Those are genes switched on in plants where there's no endophytic trichoderma associated with the plant. So unique genes that are only switched on in the plant when there's no endophytic coach involved. Then the moment you infect the plant with the endophytic trichoderma, there's a new set of genes. It's maybe not as visual on this photo, but we use software to do the analysis. And there's literally hundreds of genes being switched on in response to this endophytic coach. And then the yellow dots are just what we call the household genes, just the general genes involved in general functions inside the plant. So the endophytic trichoderma activates the genetics. So we know that they switch on genes involved in the utilization of nutrients. The genes involved in how the plant will handle stress. Remember I said these microbes are responsible to keep the plant from stressing, because otherwise the first supply of carbon that's turned down is the one to the rhizosphere. And then also genes involved in how the plant will resist disease or infection. And then all the genes involved in energy metabolism, the energy economy of the plant, or the productivity of the plant that will eventually give you yield. And as I mentioned before, also genes involved in the quality of the end product, for example, antioxidants. So the endophytic trichoderma and the healthy rhizosphere really make the most of your genetics, your plant's genetics. So these microbes are involved in switching on genes, making the most of the genetics of that plant. So in this photo, you can see it looks like 2 different cultivars. So this is a Monsanto 8040 corn cultivar, and you can see where the healthy, the endophytic trichoderma and the healthy rhizosphere has been established in the picture on the right, how the vascular tissue is so prominent and well developed compared to the control on the left. Okay, thank you very much. This is the part where you was supposed to come in.
Okay, well, thank you, Ronelle, for persevering through my telephone. We do have a couple of questions, so I'll just run through them. I've just got to get some feeling back into my hand after holding onto that phone for half an hour. Okay, the first one is, yeah, so what experience is there with half rate, I guess 2 applications, half rate applications, and in particular the questions is about onions, where there's been some good results with multiple applications. Oh, sorry, 2 half rate applications rather than 1 full rate, I think is the question. Yeah, Ronelle, can you answer that question for me?
Yes, we obviously with every crop that we test the endophytic trichoderma strains on for infection, we do different application rates. So it also depends on the soil health situation. So in a good soil health environment, a half application gives you the same result as a full application in a situation where there's more dominant groups and soil pathogens. So the recommended rate is usually based on the majority of trial results that gave us consistent results. So we know, for example, if you go below 10,000 endophytic trichoderma spores per plant, you start seeing uneven results, uneven infection and uneven results. So we never recommend going below that threshold of 10,000 spores per plant. Multiple applications, I think, is relevant for especially crops that has a dormant stage, and we know the endophytic trichoderma infects through fresh roots. So the moment the success of infection actually depends on that fresh roots being there and getting the endophytic trichoderma inside the plant. So in that case, it's important to have the timing right to infect the trichoderma on fresh roots, and then you can, you should immediately, within a week or two, there are usually a visual response that you can see that it's successful.
Okay, thanks, Ronelle. I've got another question, which is a very good segue from what you just talked about. So it's from Harry. He's asked about using Sabel X in ginger rhizome at planting, so instead of after the roots have started to emerge. So maybe you can just comment on that. It sounds like an extension of what you just talked about. Yeah, so ginger, we had a very interesting trial on ginger the past season. A big ginger farmer that had very bad problems with Fusarium. And I think if there's pathogen problems, obviously any time to introduce trichoderma is a good time, just to start controlling the dominant groups. But the best infection we see when there's fresh roots. So for making the most of your genetics, I think fresh roots are definitely an important stage to get that, to make sure that endophyte is inside and getting the best team together in the rhizosphere. Whereas the moment you are doing biological warfare, I think as soon as you see disease symptoms, obviously it's a good time to come in with trichoderma or a bacillus or whatever you use to really just address the problem of the dominant groups. Okay, thank you. There's a couple of questions that may have been asked during Neil's presentation. I think you may have answered, but I'll run through them quickly. So, Ronelle, application of Sabel X during or if there's a threat of a frost, so sort of -2, -3 degrees. So we're not talking about frozen soil, I don't think, but just an overnight frost. So the risk is definitely not on the trichoderma side. So the trichoderma spores, if it's too cool, they won't germinate. So the moment the conditions are favourable, so trichoderma spores will germinate anything from about 10 degrees Celsius and start infecting. So the moment the soil temperatures are below that, you will just add dormant trichoderma spores lying there until the conditions are favourable for germination and infection. So soil temperatures below 10 degrees Celsius usually we don't see a lot of infection. The moment the soil temperatures go above that, you see the trichoderma spores germinating and infecting the plants. Okay, so they can be applied, but they'll sit there and do nothing by the sound of it. Yes, exactly. Okay, thank you. So does trichoderma survive over the dormant period of a tree crop? Okay, I think you've just answered that one about applications, repeat applications. So another question about the A-team in the trials, where you used the beneficial A-teams. Which groups of microbes were involved in that? So we added a bacillus. We added bacillus specifically involved in suppressing nematodes. We added the Azoospermillum brasiliensis. We added, I must actually go back to so we identified the functional groups and picked in our database of microbes that we know this one suppresses fungal pathogens. So for example, a streptomyces, we added streptomyces. And I think there was a second bacillus or a pseudomonas as well. I think the pseudomonas for solubilizing the phosphates. Okay, thank you. Thank you. So Yeah, mycorrhiza. So this question is about what's the impact of Sabel X and the rhizosphere have on mycorrhizal activity. So, AMF.
Yeah, that is interesting. So, some of the endophytic trichoderma, every time you infect a plant with that specific strain of endophyte, we see a mycorrhiza establishes with it. So it obviously sends out a signal that a mycorrhiza also responds to and co-establishes in the rhizosphere. So it's not in all the strains that we see that collaboration between the mycorrhiza and the trichoderma, but there's definitely some of the endophytic strains, for example, in Sabel X Rhizosphere, that we could see there's a co-establishment of mycorrhiza and endophytic trichoderma, and actually on all the trial sites. So every time you infect the plants with that specific strain of trichoderma, you see mycorrhiza co-establishes with it. So I assume it's a team member that works very well with that specific endophytic coach.
Thanks. I missed that. I was focusing on something else. Do you does it matter the order that they're applied in terms of mycorrhiza first or trichoderma first?
I think because we know the endophytic trichoderma is very aggressive, it doesn't wait for a signal from the plant, it infects the plant. So you can get the endophytic trichoderma in within 48 hours. The mycorrhiza is actually dependent on signals. So they wait in the soil and they wait for the signal from the plant or from the endophyte. So it can take weeks for a mycorrhiza to infect. So my logic tells me to first infect the endophytic trichoderma just because you want that protection from within 48 hours. And then the mycorrhiza will either infect by itself or you can apply it or whatever your program is. But I would say definitely the endophyte that's very aggressive in its infection is the first one to be infected. Okay,
thank you for that. Okay, this is a question for Neil, and it's about stone fruit, and I just can't pronounce this. So.
Botrosphere, Steve. Botrosphere.
Okay, can you answer the question?
So I don't exactly know as much about botrosphere and stone fruit, but botrosphere in subtropical tree crops is basically a vascular tissue death inside the tree. So I would have thought anything that is going to strengthen the vascular tissue from within the root system, such as Sabel X endophyte, should then make it harder for other fungal pathogens such as botrosphere to then get into the vascular tissue to do its job. Would you comment different to that, Renelle?
No, I definitely agree. I mean, that's where we come in with the nibblers and the killers. So all the pathogens that has a mode of blocking vascular tissue responds very well if you have a beneficial endophyte inside the plant. So that beneficial endophyte definitely keeps out the pathogens and prevent them from blocking the vascular tissue. So yeah, definitely you will see a response.
Yeah, even though botrosphere is aerial, not soil.
So it's coming from the soil. It's infected through the soil.
No, botrosphere is aerial infection.
Okay. I think, Neil, obviously it would be best to test it, but I think just because you strengthen the general immunity of the plant by switching on those genes and giving the plant a better response towards stress, you will see some degree of protection. So to what degree, I mean, it's obviously necessary to test that in trials.
Okay, thanks, Renelle and Neil. Caleb has a question about using in furrow with potatoes with chemicals like Amistar. And will those spores find their way to the roots once the potatoes sprout, or as a seed dressing?
Yeah, we do seed dressing and in furrow application on potatoes in South Africa. So both is successful. And we also have a compatibility list that we can share with the Australian team. I think it's about 4 pages now. So we do compatibility. So we actually incubate the inner fitted trichoderma with a specific chemical like Amistar for 48 hours, because that we know that's the window of opportunity for the inner fitted trichoderma to get in and form that relationship with the plant or colonize the seed or whatever the situation is. And then we look at if the trichoderma is damaged by that incubation period. And there's very few chemicals. It's mostly the nematicides or the insecticides that are not on the list that you can't apply it together. You can apply it without that, outside of that 48-hour window, but not at planting and in a mix together with it.
Okay, thanks, Ronelle. Yeah, so Les had a question which sounds fairly similar to one you just answered, which was about, is there any biological antagonism between the trichoderma and the mycorrhiza? So I think it's a slightly different spin, but yeah, is there anything else you want to add to that?
No, I don't think there's. The microbes sort themselves out. So definitely, if the signal is there for a phosphate solubilizer, because most mycorrhizas are phosphate solubilizers and also provide the plant with phosphate. So if there's a specific need for that and the signal is sent out and the mycorrhiza responds to that signal, I mean, obviously it's a beneficial synergism that is established. Okay,
thank you. Rob's got a question here about long-term soil crops like greenhouse crops, tomatoes, cabbage and eggplants that might be in the ground for 8 or 9 months. How many applications would you normally recommend?
I think it depends on the disease pressure in that specific situation. If there's a problem with disease pressure, a reapplication is definitely favorable, or you will definitely see a result with a reapplication. So it's really a matter of disease pressure, what the specific situation is, and also the plant stress. So some of the farmers that are planting in greenhouses, a specific time of the year that disease pressure is very high, they reapply. Whereas during the low risk times of the year, they don't apply the Sabel X hort. But in the higher risk times, they apply like on a weekly or monthly basis, depending on the crop.
Yeah. Okay. Yeah. Look, and I know we've had some growers with real problems growing baby leaf spinach in summer, where they will apply Sabel X every week in a 4-week crop, just to make sure that they get a result that they're looking for. Okay, so Dev asked about a copy of the presentation. Yes, we will probably, given the way that it's happened, we'll probably edit it to make sure that it's in a form that everybody can have a look at, but that shouldn't take more than 2 or 3 days to get that done. So I think there was one other question there that from Harry that I missed along the way.
Yeah, shelf life, Steve.
What was the question?
They've got stock about a year old, so he was wanting to know what was the shelf life of Sabel X.
It's about 18 months, but it has an expiry date on the pack.
That's right.
And if we want to go outside of that, we'll need Ronelle to do some tests on that batch to let us know whether it's still viable or not. I think that's right, isn't it, Ronelle?
Yeah, the shelf life is 18 months. And also, I mean, microbes are sensitive towards temperature fluctuation. So the more constant you keep the trichoderma spores or whatever microbe you're using, the longer the shelf life will automatically be. So we go for worst case scenario that there will be some events that the product are exposed to some temperature fluctuation. So we build that into the product formulation. But yeah, the standard shelf life that we are comfortable with is 18 months.
Okay, excellent. Thank you. Just coming down to last couple of questions. So soil fumigation. So a lot of soil fumigation done prior to planting with annual horticultural crops. Would you normally advocate an application of the Sabel X Trichoderma together with an A team on every crop in those sort of circumstances?
Yes, definitely. We've seen really good results after soil fumigation, introducing the coach and its beneficial team members after that. So there's definitely very good results that we've got with the Sabel X and some other beneficial team members after a soil fumigation event.
Yeah, okay, thank you. There's a question here about naturally occurring organisms. When you've mentioned naturally occurring organisms, I think, well, you can answer that, but I think you were just saying what's in the soil naturally without adding anything extra. Is that right? Sorry, Renelle, you there?
Yes, sorry. So naturally occurring is obviously, I mean, it will depend on your soil health situation. So the practices, what is your practice of, what do you do with crop residue? Do you stimulate microbial activity in your soil? So obviously the better your soil practices or soil health practices, the larger the diversity and the more natural occurring beneficial microbes there will be. So in a situation where there's no focus on that, there's no crop residue stimulating microbial activity or whatever soil applications that stimulate microbial activity, there will be a tendency for more dominant groups like pathogen groups, less diversity. So your natural occurring microbes, beneficial microbes anyway, will have less of it, but will not be in an optimal condition or in an optimal state to get the best team really in the rhizosphere.
Okay, so replace them if there's been a fumigation event. That sounds perfect.
Definitely.
Okay, so one last question, and we've got from Andrew. What was the application rate of Sabel X in the Lucerne yield demo that was this applied as a post-emergent spray, or was it done at planting? So there's two parts, or a few parts of that. It was what was the application rate, and is that done at planting with the seed or as a post-emergent spray? And I can't remember whether that was your trial result or Neil's.
That was.
Application in South Africa is a dry seed treatment at planting, so 60 grams per 25 kilograms of seed, I think. Yep. That the standard application, the dry seed treatment at planting. So you want to establish that endophytic trichoderma in the beginning of that plant's life, when the rhizosphere is establishing. Okay,
so that trial would have been done as a seed treatment then, by the sound of it.
Yes, correct.
Okay, look, Renelle, thank you very much for persevering. Thank you, Neil, for your presentation. Thank you to our attendees for persevering with us as well, given our technical hiccups along the way. There's certainly, I think Rob's summed it up in his note here. Thank you. Great to sit in on this, highly informative. And I think personally from my side, it's this signalling between microbes and plants is a relatively new science from my perspective. So it does add a heck of a lot to understand, to help me understand why some beneficial microbes work better than others, and in some circumstances they don't. And it would seem to me that when they don't have all the pieces of the puzzle in place. So thank you very much for attending. That is the end of our presentation. Really appreciate you all joining us, and look forward to hopefully being able to see you all soon. Thank you very much.
Thank you all. Thank you, Steve.